Imagine a city that has just been struck by a massive earthquake.
Oh, wow. Okay, setting the scene.
Right. So buildings are compromised, the infrastructure's failing, and the whole emergency response system is just fully activated. Sirens are blaring, and emergency vehicles are rushing in from, well, every direction.
Sounds like total chaos.
Exactly. But imagine that instead of navigating through physical rubble on city streets, these vehicles are actually navigating the microscopic landscape of the human nervous system.
Okay, I see where you're going with this.
Yeah, and what if we could design these medical delivery vehicles to exclusively drop their payloads on the exact cells that are actively responding to the injury? Like completely ignoring the perfectly healthy cells located just a fraction of a millimeter away.
I mean, that sounds like pure science fiction.
It really does. But thanks to a brand new 2025 paper published in Nature Neuroscience, this is actually happening right now in the lab.
It is, yeah. It's an incredible breakthrough.
So today, our mission is to uncover the genetic regulatory code that makes this possible.
Yeah.
We are pulling apart a study from a team of scientists in Sweden who have cracked the code of injury-responsive enhancers in the central nervous system.
Which is just a massive leap forward.
Okay, let's unpack this. By the end of this deep dive, you will understand exactly how your DNA acts as a highly secure, location-specific switchboard during a medical emergency.
It fundamentally changes the whole strategy. I mean, the holy grail of treating central nervous system trauma, whether that's a severe spinal cord injury, a stroke, or a traumatic brain injury, has always been precision.
Right, because you don't want to damage the healthy stuff.
Exactly. The immense challenge in medicine is figuring out how to treat the damaged, inflamed tissue without causing massive, potentially lethal side effects in the surrounding healthy tissue.
Which is easier said than done, obviously.
Right. So to even begin targeting injured cells, we first have to understand what an injured cell actually looks like at the fundamental DNA level. A cell just going about its normal day looks vastly different from a cell fighting for survival in a highly toxic, inflamed environment.
I'd imagine the biological landscape is incredibly chaotic right after an injury. So how did these researchers even begin to map out what that looks like?
Well, they started by creating a highly controlled timeline. They used a mouse model of a spinal cord contusion injury.
Okay.
And they mapped the cellular environment at very specific intervals, so 1, 3, 7, and 28 days post-injury.
That's a pretty wide range.
Yeah, it allowed them to capture the entire biological narrative, you know, from the immediate, frantic, acute phase all the way into the chronic scar-forming phase. And importantly, they focused their attention on the glial cells.
The support staff of the nervous system.
Right, they are the ultimate first responders. In the central nervous system, neurons usually get all the glory because they transmit the electrical signals, but the glial cells are the infrastructure managers.
Like the cleanup crew.
Exactly. They manage the cleanup, they try to seal off the damage with scar tissue, and they regulate the immune response. And when trauma hits, these glial cells undergo a massive transformation.
So they just drop everything they're doing.
Pretty much. They abandon their day jobs and change their entire genetic state to deal with the crisis.
But wait, how does a cell know to just change its identity like that? I mean, if I think about human genetics, we always hear about genes being the blueprints for everything.
Sure, yeah.
But a blueprint just sits on a table. It doesn't build a house by itself. You know, a gene doesn't just spontaneously turn itself on and off.
And that brings us to the core mechanism driving this entire paper, which is enhancers.
Okay, enhancers.
Yeah. If a gene is the light bulb that illuminates a room, the enhancer is the smart home switch on the wall.
Oh, I like that analogy.
It's a completely separate regulatory element of your DNA that dictates when a gene turns on, in which specific cells it turns on, and exactly how much product it needs to churn out.
So the researchers had to find these switches.
Right. And to find these switches during a spinal cord injury, the researchers utilized a brilliantly complex dual profiling method on individual single cells. First, they ran something called RNA sequencing.
Which does what, exactly?
Well, RNA-seq essentially takes a snapshot of the factory floor, showing them exactly which genes are currently turned on and actively producing transcripts.
So that tells you what the cell is actually doing in real time.
Exactly.
But if they already know what the cell is doing, why did they need a dual profiling method? Like, what was the second step for?
Good question. They simultaneously ran a process called ATAC-seq that stands for Assay for Transposase Accessible Chromatin.
That is quite a mouthful.
It really is. But basically, if RNA-seq tells you what the cell is printing, ATAC-seq tells you which parts of the physical DNA molecule are unspooled, open, and physically accessible to the cell's machinery.
Okay, let me see if I have the visual right for this. Because our DNA is usually wound up super tight inside the nucleus, right?
Right. Very tightly.
Like a tangled ball of yarn. And if a section of that DNA yarn is tightly wound, the cell's machinery just cannot read the instructions hidden inside. It's closed for business.
Yep, that's exactly right.
So ATAC-seq is basically a tool that goes in and molecularly tags the open, readable sections of the yarn.
That is a perfect way to visualize it. It uses an enzyme that literally slips into the open, unpacked regions of DNA and tags them.
Got it.
So by layering these two massive data sets over each other, the open DNA and the active genes, they made a staggering discovery.
Which was?
They mapped nearly 28,000 highly specific genomic regions that were tightly closed, completely invisible, and utterly silent in a healthy mouse.
28,000. Wow.
Yeah. But the moment the spinal cord injury happened, those 28,000 specific sections of DNA physically popped open and became active. They named these regions injury-responsive enhancers, or IRENs.
Wait, I really want to pause on the mechanics of that. Are you saying that these thousands of emergency switches just sit there in the genome, completely turned off and hidden in that tight ball of yarn for a cell's entire life?
Yes.
Just waiting for a specific physical trauma to happen.
Exactly. The genome actually physically remodels itself in response to the toxic environment of the injury.
That is wild.
The chromatin unspools, revealing emergency instructions that were hiding in the dark the whole time. It's basically a dormant, hardwired survival program waiting for a worst-case scenario.
So if I'm tracking the logic here, we've got this massive network of glial cells completely transforming their function after an injury. And this transformation is controlled by these hidden DNA switches, the IRENs, that physically pop open only when trauma occurs.
You nailed it.
Think about your own DNA for a second, right? The idea that you have an entirely separate, heavily guarded set of genetic instructions exclusively reserved for surviving an emergency is just wild.
It really reframes how we think about our biology.
But, having 28,000 switches is one thing. How do we actually know what flips them?
And that became the next massive hurdle for the researchers. You have a database of 28,000 open DNA sequences. But to a human, it's just an endless spreadsheet of A, C, T, and G nucleotides.
Just random static.
Exactly. Finding the switches doesn't mean you can read the trigger code. They needed to find the underlying syntax that tells those switches to open.
And you cannot exactly hand a grad student a spreadsheet with billions of genetic letters and ask them to find the grammatical rules. Where do you even begin to translate that?
Well, you bring in deep learning. The team utilized a specialized convolutional neural network called CROM-BKNet.
Okay, an AI.
Right. And the choice of a convolutional neural network, or CNN, is incredibly important here. CNNs are often used in advanced image and facial recognition.
Oh, like when your phone recognizes your face.
Yeah, exactly. They don't just look for a generic nose and two generic eyes. They map the exact spatial relationship and the distance between those features. So the AI was trained to look at the raw DNA sequences of these highly specific IRENs and predict whether the DNA would be physically open or closed, based purely on the arrangement of the letters.
Here's where it gets really interesting, because the AI didn't just memorize the sequences of letters, did it? It actually learned the spatial grammar of the injury response. And what the algorithm revealed is that the cell uses a highly secure login protocol to prevent accidental activation.
Yeah, the deep learning model uncovered a cooperative syntax. For an injury-responsive enhancer to activate, it requires two distinct transcription factors to bind to it at the exact same time.
And transcription factors are basically just molecular keys, right?
Yeah, they're the molecular keys that lock into the DNA switch. The first key the AI identified is a lineage-specific factor, for instance, a molecule called SOX9.
SOX9. Right.
The presence of SOX9 essentially announces to the dytome, I am an astrocyte.
So it functions as the username. It establishes the cellular identity.
Exactly. But the username alone cannot open the emergency enhancer. The second key the AI found is a stimulus-responsive factor.
Okay, what's that one?
They found a massive enrichment of a factor called AP1 across these specific enhancers. AP1 is a molecule that floods the cellular environment specifically when there is stress, damage, or inflammation.
Wow.
Yeah, it essentially tells the DNA there is a severe injury happening right now.
It is literal two-factor authentication. It's a biological 2FA system.
It really is.
You cannot log into this genetic emergency program with just your username, meaning just being a healthy astrocyte isn't enough to trigger the switch.
Right.
And you cannot log in with just the password, meaning an injury signal floating around in the wrong type of cell, like a liver cell, won't trigger it either. You need the astrocyte username and the injury password binding together in the same physical space to flip that genetic switch.
It's incredible. Nature evolved this brilliant failsafe to encode cell state specificity.
To keep things from misfiring.
Exactly. It forces the simultaneous recruitment of broad injury signals alongside very strict cell identity programs to prevent extreme immune or scarring responses from misfiring in healthy brain tissue.
Okay, so just to recap for a second, scientists used a deep learning neural network to read the raw DNA of these hidden switches, and they decoded this biological 2FA system requiring both identity and injury molecules.
Yep, that's the summary.
Which raises a fascinating idea. If we now know the login credentials for a specific disease state, what is stopping us from hijacking that exact same login screen to send in our own biological software?
Nothing is stopping us. And that question drove the researchers to the next critical phase, which is in vivo validation, meaning live animal testing.
Okay, so out of the computer and into the real world.
Right. They moved from computer models into biological engineering. They took these newly discovered IREN sequences and packed them into adeno-associated viruses, or AAVs.
What's an AAV?
AAVs are essentially hollowed-out virus shells commonly used as delivery trucks in gene therapy because they are safe and efficient at entering human cells. Ah, okay. Delivery trucks.
Yeah. But instead of letting the virus deliver its payload indiscriminately, they genetically engineered the IREN sequence onto the payload, basically acting as a lock on the cargo doors.
Oh, that's clever.
For this experiment, the payload itself was just a glowing fluorescent tag, so they could track exactly where it opened.
The concept of the delivery truck makes total sense. But how did they administer it? I would assume they injected the virus directly into the site of the spinal cord injury to make sure it got there.
See, that's the crazy part. They actually injected the viral vectors systemically into the entire bloodstream of the mice.
Wait, into the whole bloodstream?
Yes. The delivery trucks were circulating everywhere. Through the heart, the liver, the healthy hemispheres of the brain, and finally the injured spinal cord.
Wow.
And out of the different enhancers they screened, one in particular emerged as the star player. They called it IRN5.
IRN5.
Yeah. This specific enhancer is naturally linked to a gene called CD44, which is heavily upregulated when tissue responds to severe damage. I am still a bit confused on the targeting aspect, though. If you inject a viral vector into the entire circulatory system, it's entering millions of perfectly healthy cells all over the body.
Sure. Yeah.
Why doesn't it just drop the payload everywhere? How does the virus know it's at the scene of the accident?
Well, the virus doesn't actually know. The targeting is entirely handled by the two-factor authentication of the IRN5 lock on the cargo door.
Oh.
Yeah. When the IRN5 virus entered a healthy astrocyte miles away in the uninjured cortex of the brain, the cell provided the SOX9 username. It had the correct astrocyte identity.
Right.
But because there was no trauma in that part of the brain, it completely lacked the AP1 emergency password.
So the 2FA failed.
Exactly. The 2FA failed. The virus remained locked, silent, and completely invisible. The glowing tag never activated.
That is an incredibly elegant mechanism.
Yeah.
It uses the body's own localized crisis as the trigger.
It really does. The glowing tag only turned on in the reactive astrocytes directly bordering the lesion site in the spinal cord, achieving an 85% specificity rate.
85%. That's amazing.
And it gets better. To ensure it was robust, they also tested it in a traumatic brain injury model, basically a physical stab wound to the cortex.
Okay.
In the TBI model, the systemic IRN5 virus successfully labeled 98% of the astrocytes right along the border of the brain injury, while leaving the perfectly healthy opposite hemisphere of the brain completely dark.
98%. It is the ultimate biological smart bomb. It floats harmlessly through the vascular system until it finds the exact coordinates of the disaster zone.
It's unprecedented precision.
So we've established that scientists can build custom viral delivery trucks using IRENs as the cargo locks, successfully delivering a payload exclusively to injured cells. But, in science, seeing a machine work is only half the proof.
Oh, absolutely.
Sometimes you have to intentionally break the machine to prove you actually understand how the gears fit together. So how did they test the integrity of this 2FA code?
Well, they utilized a technique called in silico mutagenesis. They used their CROMB-PNet AI model to simulate very specific mutations in the IRN5 DNA sequence, manufactured those intentionally broken viruses, and introduced them into live animals.
Break the code to see what happens.
Right. The first test was simple. They surgically deleted the AP1 site from the DNA. They deleted the injury password.
So we know taking out the injury signal kills the response. The switch just stays dead because the payload can never verify the trauma.
Yep.
But what happens if we mess with the other half of the code? What if they delete the SOX9 astrocyte username?
When they deleted the SOX9 site, the enhancer lost its precision entirely. It still sensed the injury via the AP1 password, but it forgot which cells it was supposed to be operating in.
Oh, so it just went rogue.
Basically, yeah. It started activating indiscriminately in the wrong cell types near the lesion. The dual lock system really requires both pillars, but the most profound experiment was when they tested the so-called filler DNA.
Ah, yes. This is the part that genuinely blew my mind when I read the methodology. We always picture DNA as this incredibly delicate, highly specific blueprint where every single letter matters. How did they manipulate the filler DNA?
They went in and synthetically shuffled all the surrounding DNA base pairs inside the enhancer.
Wait, really?
Yeah, every single genetic letter that wasn't the AP1 password or the SOX9 username got completely scrambled. They randomized the background sequence.
And what happened?
The result: the virus still perfectly navigated to the injured cells and activated with precision.
Let me make sure I'm wrapping my head around that. It is like taking a complex sentence, scrambling every single letter of every single word into complete gibberish, except for the main noun and the main verb. And the cell somehow still perfectly reads the command.
That is the perfect analogy, and it completely flips our traditional understanding of genetic architecture.
It's hard to even comprehend.
Right, because for decades, the assumption in evolutionary biology was that all that extra DNA surrounding a functional motif was critical. Rigid scaffolding built up over millions of years of evolution.
We thought it was all load-bearing.
Exactly. We assumed the specific sequence of the background was vital for the DNA to fold correctly. But this experiment proves that the strict syntax, the exact spacing, orientation, and presence of those specific molecular anchors is the undisputed master key.
So the rest of it really is just filler.
Pretty much. All that surrounding evolutionary DNA clutter is largely irrelevant as long as the 2FA pillars are in the precise spatial position. The cellular machinery doesn't read the whole paragraph. It just scans for the specific grammar.
The fact that the foundational building blocks of life are that modular and programmable is staggering. It really does behave like computer code. You can pull out variables, delete functions, and as long as the core syntax is intact, the program runs.
It's a bioengineer's dream.
So what does this all mean for us? We've been talking a lot about glowing fluorescent tags, which is obviously how scientists visualize the success of these experiments in mice. But why does a 2025 Nature Neuroscience paper about glowing mouse cells matter to the future of human medicine?
Well, because you don't have to pack a glowing tag into that viral delivery truck. The tag is just a proof of concept.
Right.
You can pack that engineered virus with profound therapeutics. Reactive astrocytes, the specific cells targeted by the IRN5 enhancer, are deeply involved in the progression of spinal cord injuries.
Which we know are devastating.
Right. But they're also the major players in the destructive aftermath of strokes, in the secondary damage of traumatic brain injuries, and in severe cascading neurodegenerative diseases.
And those are so hard to treat.
Incredibly difficult. Right now, treating these catastrophic conditions is basically a blunt instrument. Any drug powerful enough to chemically alter the behavior of a damaged, inflammatory astrocyte is also going to wreak havoc on the healthy astrocytes that your brain desperately needs to maintain normal cognitive function.
So the side effects are just too severe.
Exactly. Systemic side effects limit how aggressively we can treat these traumas.
But by utilizing these newly mapped injury-responsive enhancers, we bypass the side effect problem entirely.
We do.
Imagine a clinical future where immediately following a severe car crash, stroke, or a major concussive event, a patient in the ICU is given a simple IV drip.
Just a standard IV.
Yeah, and that IV contains billions of viral delivery vehicles equipped with an iron lock. They flow harmlessly through the patient's entire circulatory system. They pass through the healthy heart tissue, the liver, the intact lobes of the brain, completely inert.
Just waiting for the 2FA.
Exactly. But the second they cross into the precise millimeter of the brain or spinal cord where the physical trauma occurred, the biological 2FA clicks into place. The DNA unspools, the switch flips, and the vehicles deploy a payload of healing proteins, regenerative growth factors, or anti-inflammatory agents perfectly on target, with absolute zero collateral damage to the rest of the nervous system.
It represents the ultimate realization of precision medicine. We are no longer relying on flooding the entire body with toxic drugs and hoping the benefits outweigh the side effects.
It's a completely new paradigm.
It really is. We are co-opting the body's own ancient, hardwired emergency response syntax to deploy medicine exactly when, where, and only as long as it is needed, right at the single-cell level.
It brings us right back to those emergency vehicles from our earthquake scenario, effortlessly bypassing the quiet, healthy suburbs and driving straight into the smoking rubble to deliver aid exactly where the signal flare is coming from.
That's exactly what's happening.
Well, we have covered a massive amount of ground today, from the physical unspooling of hidden DNA to deep learning neural networks decoding the cooperative grammar of our fundamental survival mechanisms. But before we wrap up, I want to leave you with something deeper to chew on as you go about your day.
Let's hear it.
Today, we have entirely focused on the biological two-factor authentication code for injury, the IRENs, the emergency switches. But what about the hidden regulatory code for all the other complex states our cells exist in?
Oh, that's a great question.
If your genome has 28,000 hidden switches just waiting to be physically flipped by trauma, what is the exact enhancer syntax for forming a brand new long-term memory? What is the specific genetic password your brain requires to enter deep, restorative sleep?
Or learning to ride a bike, right?
Exactly. Or the enhancer syntax for learning a complex new motor skill, like playing the piano. If our shifting dynamic cellular states are just waiting for the right molecular keys to be decoded by AI, how much of your everyday human experience is simply a programmable genetic switch sitting quietly in the dark, just waiting for the right two molecules to bind together and turn the lights on?
Something to think about for sure.
Keep exploring that idea. Thanks for joining us on this deep dive.